won't save. So, with that said, let's talk about the lymphatic and the immune systems. So, we had talked briefly about circulatory systems earlier. Actually, we will talk briefly about circulatory systems in this class. We have two main branches of our circulatory systems. The one that most people focus on is the cardiovascular system, so the heart pumping blood. The other branch of our circulatory system that not many people think about is the lymphatic system. So today we're going to focus on the lymphatic system. Our lymphatic system is very, very closely tied to our immune system. In human physiology and in microbiome, we spend a lot more time talking about the immune system. This presentation is mostly going to focus on structural anatomy of the lymphatic system. If we look at our lymphatic system, its primary purpose is to maintain fluid balance. We have leaky capillaries all over our bodies. And this is a good thing. We need liquid to leak out of our blood capillaries and go into the interstitial fluid and surround the cells. This is how we transfer nutrients from our blood to the cells, and then from the cells back to the blood by having this extracellular fluid. This fluid that is outside of the capillaries and outside of the cells. So all the cells of our body are surrounded by liquid, and we need the right amount of liquid surrounding the cells of our body. And this liquid, this extracellular fluid, is outside of the blood vessels and it's outside of the cells. Sometimes it's referred to as interstitial fluid because it's in between the cells. And I'm willing to bet that you've seen somebody who's had too much interstitial fluid in one of their appendages. Usually it's one of the legs that gets too much interstitial fluid. What's that called?
Edema.
Edema, particularly lymphedema. If it's just a regular edema, normally that's referred to as bruising.
Oh.
Or bleeding in the skin. So if we have too much extracellular fluid build up in the appendage or in the extracellular space, they'll have lymphedema. It manifests itself in a patient. The secondary function of our lymphatic system and primary function of the immune system is to protect us from infection and disease. Our lymphatic system aids us with our immunity. All of that liquid that leaks out of our capillaries that goes to the interstitial space and becomes interstitial fluid is going to be sucked up and filtered. Have you ever worked at a public pool or cleaned a pool filter?
No, but I can imagine.
Oh, they are nasty. So if you look at the water at a public pool, you look at it and it seems fairly clean, but there's a low concentration of Band-Aids and body hair and other bits and pieces of stuff. And then all that pool water is going to be sucked up and filtered, and all of the contaminants in that pool water are concentrated at the site of filtering. In the same way, we'll take that extracellular fluid, suck it up into the lymphatic system, and concentrate any impurities, any potential pathogens in our lymph nodes. Those immune cells, generally speaking, are going to be waiting in the lymph nodes, and they'll be constantly screening the materials that are filtered out of our lymphatic fluid. Something else our lymphatic system does, particularly in the small intestine, is it aids us with the absorption of body or fat that we eat. The sugar that we eat, the protein, and the nucleic acids that we eat are all absorbed directly into the bloodstream. And this is because those biomolecules, the carbohydrates, the proteins, the nucleic acids, have polar side chains and they're hydrophilic. They mix well with water. Fat doesn't mix well with water. So it's really hard to absorb fat directly into the bloodstream. Instead, we'll absorb those fats, those lipids, into small extensions known as lacteals. And those lipids that are absorbed into our lymphatic system are then processed and sent to the other parts of the body. And then here's the part that most people focus on with our lymphatic system: fluid recovery. Our lymphatic system helps us absorb liquid that escaped from the bloodstream and dumps that liquid back into the bloodstream. So, Izzy, any questions so far? By the way, thank you so much for coming.
Thank you for not going to attend, said I was still working.
So let's define some terms here. we have interstitial fluid and lymphatic fluid, or sometimes referred to as lymph. So when you see this term lymph, that's a synonym for lymphatic fluid. And the difference between interstitial fluid and lymphatic fluid is location, location, location. Interstitial fluid is the liquid outside of the bloodstream and outside of the lymphatic system and outside of the cells. It's that liquid around the cells. The moment that interstitial fluid is sucked up into a lymphatic capillary, we arbitrarily rename it lymphatic fluid, even though its chemical composition is, for all intents and purposes, identical. Now, that lymphatic cap liquid, just like our interstitial fluid, is going to be clear. It's colorless. It's very, very similar to plasma. Now, when we say plasma, what liquid are we referring to? Blood plasma. Excellent. So this lymphatic fluid is almost identical to blood plasma because this lymphatic fluid is essentially interstitial fluid, which is also almost identical to blood plasma. So we don't have proteins, or excuse me, we don't have a lot of proteins in our lymphatic fluid. Most of those soluble proteins are going to be stuck in the bloodstream and are not able to leak out of the bloodstream. There's very, very few places in the body where we're supposed to have our blood plasma proteins leak out into the interstitial spaces. So because most of the proteins stay in the blood plasma, our extracellular fluid or interstitial fluid has very little protein in it, and consequently, our lymphatic fluid will also have very little protein in it. That fluid is going to be transported in a series of tubes known as lymphatic capillaries. Something that's unique about these lymphatic capillaries is they don't make a continuous circulatory route. They're more of a dead-end street. They are completely closed off at one end. To keep them in place, to keep them from moving around inappropriately, we're going to anchor them to the tissues of our body with small filaments of proteins that will kind of act like tent posts or tent pegs, anchoring these closed lymphatic capillaries in place. Now, just because they're closed at one end doesn't mean that they are impermeable to liquid. If they were impermeable to liquid, we would not be able to take that extra interstitial fluid and dump it into the lymphatic capillaries. Our lymphatic capillaries are going to have little gaps in them. The endothelial cells that make them up are very loosely overlapped. This allows the lymphatic capillary, to function with very weak one-way valves, or very poor one-way valves in them. So as we have pressure building up in the extracellular environment, that's going to force interstitial fluid through the loosely overlapped endothelial cells into the lymphatic capillary. So if you're working with a patient that has lots of extra fluid in an appendage that has lymphedema, massage is one of the best treatment methods for lymphedema because squeezing or putting pressure on the appendage where there's too much extracellular fluid increases the pressure on that extracellular fluid and forces it into their lymphatic capillaries. One of the big side effects of radical mastectomies, which are typically only done for women that have breast cancer, is that the process of the radical mastectomy not only removes the breast, but also removes a lot of the lymphatic tissues, or lymph nodes and lymphatic capillaries. And the side effect of that radical mastectomy is lymphedema in the corresponding arm on the side that it was performed on. I don't know about you, Izzy, but when I was younger, I always wondered to myself, why is breast cancer such a big deal? Why does it kill so many people?
Cut them off, you're good.
Yeah, I mean, it's not like you need it to be alive.
Right, I did.
The reason breast cancer is so deadly is because there are lots and lots of lymphatic capillaries in the breasts. And it's very, very easy for those cancerous cells to get into the lymphatic system and then rapidly transport all over the body. but you are right. The breast itself is not essential for living. So here's some of those structures we've talked about. We have this closed end of the lymphatic capillary. Notice how it's not open. But we're going to have, if we look right here, loosely overlapping endothelial cells. And these loosely overlapping endothelial cells are what function as those weak one-way valves that allow interstitial fluid to get pushed into the lymphatic capillary. And then we also have these anchoring filaments that help hold the lymphatic capillaries in place.
Well, lymphatic what?
Capillaries in place. On this figure, they use the term tissue fluid. I have been saying interstitial fluid. They're the same thing. It's that liquid in the tissues outside of the cells, outside of the bloodstream, outside of the lymphatic capillaries. Interstitial fluid. I suppose I could say tissue juice, but that's not specific enough. Certainly not academic sounding enough.
Right.
Kind of like when we studied the eye structures, we can't say eyelids, we have to say palpebrae. So if we look at these lymphatic vessels, they have almost an identical composition to our blood vessels. There's 3 layers to them. We're going to have the tunica interna, the tunica media, and the tunica externa. This tunica interna is made up of very slippery endothelium. This endothelium in the tunica interna has a very, very low frictional coefficient. In other words, it's super slippery and liquid can flow through it pretty easily. The tunica media, just like with our blood vessels, is going to have smooth muscle in it. And if we had to compare lymphatic vessels to either arteries or veins, our lymphatic vessels would be more similar to veins than they would be to arteries. Arteries have very thick tunica medias. Veins have very thin tunica medias. Something else that a vein has that an artery does not have is the one-way valves. So if we compare the lymphatic vessel to a vein, there is going to be a lot of similarities. Just like with veins, we'll have a tunica externa. It's a very thin outer layer composed primarily of connective tissues that helps to anchor our lymphatic vessel in place. Here is a histograph of some lymphatic vessels. So in this figure, our lymphatic vessel is right here. And you can actually see the endothelium overlapping on itself on the inside, forming the one-way valve in our lymphatic vessel. Any questions, Izzy? So we have all of this extracellular fluid or interstitial fluid in our bodies. We need to get all of this extra fluid that's leaked out of our bloodstream back into the bloodstream. Ultimately, all the extra fluid is going to go back to the bloodstream and then we urinate it out or breathe it out to get rid of most of the extra fluid in our bodies. But to get it back into the bloodstream, we're going to have some connections between the lymphatic system and the bloodstream. There needs to be a point of entry. The 2 points of entry that I want to emphasize to you are the right lymphatic duct and the thoracic duct. These are the 2 spots in our body where lymphatic vessels drain their lymphatic liquid back into the cardiovascular system.
Lymphatic vessels drain.
Lymphatic fluid into the cardiovascular system. If we want to look at the direction of travel, starting at the beginning, I'll wait for you to finish writing.
Okay.
So starting at the beginning, we have number 1, lymphatic fluid traveling through lymphatic capillaries. From there, it goes to the collecting vessels. During this process of going through collecting vessels, there is going to be multiple lymph nodes filtering the lymphatic fluid. So when you see the term lymph node, I want you to think of a lymphatic fluid filter. This is the filter of our lymphatic system. And just like with those pool filters where all the nasty Band-Aids and body hair gets concentrated at the filter, in our lymph nodes we'll have bacteria and pathogens and parasites all being concentrated from the extracellular fluid into the lymph node.
If your body isn't filtering the interstitial fluid, what organ is filtering?
It's going to be primarily associated with skeletal muscle tissue. Most Usually the things that would cause lymphedema to occur are going to be a combination of factors. Obesity, inactivity, and the removal of lymph nodes are the three biggies that contribute to lymphedema occurring. Inactivity contributes or is a risk factor for lymphedema because as we move our bodies and contract our skeletal muscles, we cause localized buildups and pressure in our body. And as we have an increase in pressure, we force liquid through the lymphatic vessels and lymphatic capillaries. So one thing that's often recommended for individuals that are suffering from fluid buildup is just go for a walk. Get your body moving. The more you move your body, the more circulation you'll have in your body. It's also one of the big reasons why bed rest is generally speaking frowned upon in most situations now. Obesity contributes to lymphedema because as more and more as the body increases in volume, there is going to be more and more fluid that needs to get pumped through miles of more capillaries. And that's a lot of extra work that needs to get done. And then we had inactivity, obesity, and then removal of lymph nodes. So think back to that breast cancer example. If we remove lymph nodes and lymphatic vessels from the body, that lymphatic fluid doesn't have anywhere to flow through. So it'll start to build up and pool in the appendage. Did that answer your question?
Yes.
Excellent. So we go from lymphatic capillaries to collecting vessels. That lymph fluid, the lymphatic fluid, is filtered at the lymph nodes. And because this is where those pathogens will be concentrated, we're also going to have a lot of white blood cells waiting for us, or waiting in the lymph nodes. So we have many white blood cells hanging out in the lymph nodes, screening the lymphatic fluid as it's being filtered. The last time you were sick, what happened to your lymph nodes? They swelled up. What happened there as your lymph nodes were swelling up is you had a pathogen entering your lymph node. That pathogen was getting activated white blood cells in your lymph node, and then those white blood cells started to rapidly multiply. And as you had an increase in cell mass of white blood cells, and you have an increase in bacterial cell mass in your lymph nodes, you have more white blood cells, you have more bacteria, the lymph node swells up. Ultimately, those collecting vessels will merge at very larger lymphatic vessels known as lymphatic trunks. The lymphatic trunks merge at the collecting ducts, and our two big collecting ducts are the right lymphatic duct and the thoracic duct. I give a written description of what parts of the body are controlled or drained by the right lymphatic duct and thoracic duct, but I prefer this picture. I think this makes more sense, or it's easier to understand. Our right lymphatic duct is going to drain into the right subclavian vein. If we have damage to the right lymphatic duct, let's say the right lymphatic duct gets clogged up, so we can no longer drain lymphatic fluid out of it. If that happens, we will have fluid buildup in that portion of the body. On the flip side, we have our thoracic duct. The thoracic duct is also going to drain into a subclavian vein. But instead of draining into the right subclavian vein, this thoracic duct drains into the left subclavian vein. And it is going to take fluid from lymphatic fluid from the rest of the body. Now refresh my memory, please. What direction does blood flow in veins? Does it go to the heart or away from the heart?
Blood flow?
Yep.
Well, is it oxygenated or deoxygenated?
Irrelevant.
Oh. Which direction does blood flow to the heart? Is that what you asked?
In a vein, what direction is the blood flowing relative to the heart? To the heart or away from the heart?
To the vein, to the heart.
Bingo, to the heart. So these lymph nodes, or excuse me, these lymphatic vessels, the right lymphatic duct and the thoracic duct all drain into veins that are right next to the heart. So that fluid is immediately going to go into the heart and be distributed to the rest of the body through the cardiovascular system. 20% of the blood that comes out of the left ventricle is going to go directly to our kidneys, where we produce our urine. So from the lymphatic ducts draining into the cardiovascular system, it's a very short trip to the kidneys where we can urinate out excess fluid. So here's a figure emphasizing what we just talked about, but in a little bit more detail. We're going to have liquid leak out of our blood capillaries. So we have leaky blood capillaries. Those leaky blood capillaries have interstitial fluid build up around them. As the interstitial fluid or extracellular fluid builds up, it's going to slowly work its way into lymphatic capillaries. Those lymphatic capillaries converge and form collecting vessels, and those collecting vessels will have their lymphatic fluid filtered at a lymph node. From the lymph nodes, the lymph is going to keep flowing till it gets to a collecting duct. And from the collecting duct, the lymphatic fluid is dumped into the cardiovascular system, where some of that fluid will make its way back to a capillary, leak out of the capillary again, and continue the cycle. Any questions? Okie dokie. Here's a figure emphasizing the heavy concentration of lymphatic tissue in the breast. And this is true for both male and female breasts. In the breast, there is a very high concentration of lymphatic vessels and lymphatic capillaries. These vessels are going to wrap around laterally and go into the axillary region. That axillary region is a fancy term or technical term for the armpit area. And from there, those lymphatic vessels are going to work their way to either the right lymphatic duct or that other duct we had mentioned.
Left, or the thoracic.
The thoracic collecting duct. So here's a figure showing our thoracic collecting duct or thoracic duct, also draining into a subclavian vein. As you can see, we have lymphatic vessels and lymphatic capillaries distributed all around our bodies. And the distribution of lymphatic capillaries is going to mirror the distribution of our blood vessels. Generally speaking, in the parts of our body that have lots of fluid flow, we are going to have lots of lymphatic capillaries and lymphatic vessels to help remove the excess fluid that leaks out of the bloodstream. So let's talk about a little bit of the physiology. How can we get this lymphatic fluid to move? All fluids are going to flow from an area of high pressure to an area of low pressure. And there's two kinds of fluids in our body.
Fluids flow from high pressure to low pressure?
Yep, from high pressures to low pressures. The two kinds of fluids in our body are either going to be liquid fluids or gaseous fluids. And that second one is the one that most students find counterintuitive. Gas, like the air around us, is actually a fluid. It will flow from an area of high pressure to low pressure. And that's how we can breathe in and breathe out, by changing the pressures in our thoracic cavity. But we aren't talking about the respiratory system right now, so we'll focus mostly on the liquid lymphatic fluid. So as we increase the pressure, we'll make the lymphatic fluid to an area of low pressure. Because we don't have a specific organ dedicated to being a pump for our lymphatic system, our lymphatic fluid, generally speaking, is low pressure and low velocity or low speed. We don't have a separate part pumping lymphatic fluid in our bodies. As we have the smooth muscle in the tunica media constricting, we are going to have rhythmic contractions that move the lymphatic fluids through our lymphatic vessels. Just like with our veins, we have a skeletal muscle pump that also helps to move fluid through our lymphatic vessels. And this is one of the primary reasons why going for a walk, getting some physical activity, will speed up the rate of lymphatic flow. Getting that physical activity will make it so we can better filter our lymphatic fluid. It's one of the reasons why low levels of exercise, as opposed to no exercise, helps us fight infection. People that are on extended bed rest, or patients that have extended bed rest, generally speaking, are more susceptible to diseases and infections because they don't have their skeletal muscle pump being activated to aid the lymphatic fluid flow. We also have a thoracic pump. This thoracic pump has to do with the pressure changes in our thoracic cavity. When you inhale, you are having fluid flow into your thoracic cavity. The fluid that most of us focus on is air. So take a deep breath. You have tons of fluid rushing into your mouth, going through your windpipe into your lungs. That's because you had low pressure in your thoracic cavity when you were inhaling. This low pressure in your thoracic cavity that also causes air to flow into your thoracic cavity also causes blood and lymphatic fluids to flow into the thoracic cavity. When we inhale, we have that low pressure in the thoracic cavity. And as I mentioned before, fluids always go from areas of high pressure to low pressure. To keep the lymphatic fluid from flowing backwards, we have those one-way lymphatic valves. Once we get the lymphatic fluid close to the subclavian veins, there's something known as the Bernoulli effect. Don't bother, it's a physics term. The idea is, if you have liquids that are rapidly moving past an opening, these liquids that are rapidly moving past the opening are going to suck other liquids down into the opening. This Bernoulli effect is the reason why we need to have chimneys on our houses that go up above the roof line. If the chimney goes up above the roof line, we get a wind going across the chimney, and that creates a vacuum that sucks air up the chimney.
Are you going to put that word on the past?
Absolutely not. You do not need to know the term Bernoulli effect. So by having the rapidly flowing blood in the subclavian veins, that rapidly flowing blood creates a vacuum or an area of low pressure that sucks lymphatic fluid into the subclavian veins. And then, I've said it before, a whole bunch, I'll say it again, exercise makes it so that we have more lymphatic fluid flow. Primarily why though? Why would exercise make it so we have much quicker lymphatic flow? Which one of these mechanisms does exercise affect? Or which mechanisms would exercise affect?
The pressure, the high and low pressure.
How so? How high and low pressure? So I'm exercising a lot. What am I contracting and relaxing?
Your thoracic.
Absolutely. If I'm exercising a lot, I'm generally breathing a lot. So the thoracic pump is going to be increased. What else would be increased during exercise?
Your heart rate.
Heart rate, and if the heart rate goes up, this blood will flow even more rapidly. What else goes up during exercise?
Your muscle mass.
Not the muscle mass, but the muscle contractions in our skeletal muscles. So exercise increases skeletal muscle pump activity, it increases thoracic pump activity, it increases blood flow, and it also stretches our lymphatic vessels to increase the rate of contraction of the tunica media within the lymphatic vessel. So there are multiple reasons why exercise is great for us in terms of getting our lymphatic fluid flowing. For most of us that have the sedentary lifestyles, we are depriving ourselves of this lymphatic fluid flow. Any questions, Izzy? Okie dokie. Our lymphatic fluid tissues are not going to be highly concentrated tissues. You can't take a chunk of, or a big slice of steak and compare that to a big slice of lymphatic tissue. Skeletal muscles and most tissues of our body have highly concentrated areas of tissue, of that tissue type. Lymphatic tissue, on the other hand, is very diffuse throughout the body. There's going to be long strings or filaments of lymphatic tissue flowing through our body. Some of our lymphatic tissues are going to be in little patches or areas that are concentrated in our mucous membranes. Other lymphatic tissues are going to be interspersed in the connective tissues. So in this context, CT is connective tissues. Tissue.
Tissue. That's it.
With an S at the end. There we go. One of these areas of lymphatic tissue that's in the mucous membranes is going to be called the mucosa-associated lymphatic tissues, or MALTs. These are areas that are open to the external environment. And they sample the external environment and take the external environment and dump it directly into the lymphatic system so that we can filter it and send it to the lymph nodes. And what do we have waiting in the lymph nodes? What's waiting in the lymph nodes to sample what we filter out of the lymphatic fluid? What was that?
Did I learn that?
You did? Yes. It's part of our immune system.
Tell me.
White blood cells. So let's say you eat some chicken salad or potato salad that was sitting out at the potluck for too long and it had a very high bacterial concentration to it. As you consume that spoiled chicken salad, it goes into your digestive tract. Your digestive tract has lots of MALTs, or mucosa-associated lymphatic tissues, in them. And at these little areas that are connected to the lymphatic system, we will have that bacterium that you ate get sucked up into the lymphatic tissues and then sent to the lymph nodes for processing. If we look at our lymph nodes, these lymph nodes have very dense masses of, lymphocytes or lymphatic cells. These lymph nodes, generally speaking, are about the size of a kidney bean if they are not swollen up. Sometimes they are larger though, and if we're actively fighting an infection, the lymph node will increase in mass. Within our lymph nodes, we are going to have lots of white blood cells waiting to sample those pathogens. And this term right here, a lymphocyte, is one of the kinds of white blood cells that we're going to talk about. Izzy, can you refresh my memory, please? What's the technical term for white blood cell? For red blood cells, it's erythrocyte.
Oh.
For a white blood cell, it would be starts with an L. You're on the right track. Leukocytes. So these lymphocytes are one kind of leukocyte that we're going to talk about by the end of this presentation. In physiology, we spend a lot of time talking about lymphocytes specifically. In this class, we're going to focus on kind of a broad overview of all the leukocytes. We also have a structure known as a Pryor's patch. A Pryor's patch is found at the junction of the small and large intestine. We have very high concentrations of Pryor's patches in our appendix. Contrary to popular belief, our appendix is not a vestigial organ. The term vestigial is really a lazy term. It means we haven't figured out what the structure does yet. It doesn't mean that the structure has no function. It means we don't know what the function currently is. Can you think of any other vestigial organs or classically vestigial organs off the top of your head? There's usually one that most people mention by our posterior. The coccyx, also known as the tailbone.
We don't know what that does?
We do know what it does now. We have muscles that control our anal sphincters that connect directly to the coccyx that help keep things from flowing backwards through our rectum and anus.
We didn't know that
So that is an example of the function of the coccyx. Calling the coccyx or the tailbone a vestigial organ is a misnomer now because we know the function. We also know the function of the appendix. It has lots of Pryor's patches in it to help us sample what we've been digesting. Now, can we live without an appendix?
Yes.
Absolutely. If we lose some of our lymphatic tissues or our lymph nodes, we can still function fairly well. We have lots of redundancy in this system.
Do we know what appendix does now?
Yeah.
So a doctor says he doesn't know. That's just when he was taught way back then?
Generally speaking, that's because the doctor has been out of med school for a while.
Okay.
Now, just because we know what the function of the appendix is, doesn't necessarily mean that it's a super duper important function. Obviously, we can get by just fine without an appendix. It's when somebody has their appendix and most of their other secondary lymphatic organs removed that they start to have problems. So, let's talk about our lymphatic organs. Most of our lymphatic organs that we define as an organ are going to have specific sites in our bodies. They are going to be surrounded by connective tissues to help encapsulate them. And we break these up into two categories, primary and secondary lymphatic organs. Our primary lymphatic organs are absolutely necessary. If we have these primary lymphatic organs removed early on in our lifespan, let's say before the age of 12, we are going to be severely impaired in terms of our immune system. Our primary lymphatic organs are where we make our T cells and our B cells become immunocompetent. That's a big technical term, fancy pants term, to say that we program our T cells and our B cells to recognize foreign pathogens and to not attack the native cells of our body. Every now and again, there's a malfunction where the T cells and B cells are improperly programmed and think that something in our body does not belong in our body.
Program T cells and B cells to recognize
Pathogens, or to attack pathogens and not our own body tissues. What's it called? There's a special kind of disease when this process goes wrong and our T or B cells start to attack our own body tissues. An autoimmune disease. So when that happens, it's because our T and or B cells are gone wild. They're misfunctioning. The two primary lymphatic organs are red bone marrow and the thymus. And here's a good analogy for you. T cells are matured in the thymus. And then B cells are matured in that red bone marrow. And I have to let you know, just for the sake of being technically correct, B cells are not called B cells because they are matured in the bone marrow. That's just a nice coincidence. They're originally called B cells because they were isolated from the bursa of chickens. So that B is for avian bursa. But I like the B for bone marrow analogy. It works well. So we have our primary lymphatic organs. We have our secondary lymphatic organs as well. These are going to be organs that will include our lymph nodes, our tonsils, our spleen, and to a lesser extent, our appendix. So after we make the T and B cells and program them correctly in the thymus and the red bone marrow, those T and B cells will then move to our lymph nodes, our tonsils, and our spleen. Any questions? So let's first focus on lymph nodes. The only organ in our body that filters lymph and concentrates the pathogens in the lymph is the lymph node. Our lymph nodes have more afferent vessels than efferent vessels. And you're probably at this point thinking, what on earth is he talking about?
I remember the word this one.
Efferent means flow out of. Afferent means flow into. I always like to think of exit with an E, efferent. An efferent vessel is exiting.
Efferent flow out of?
Yes.
More efferent?
Fewer efferent than afferent vessels. So in other words, another way of thinking this, Izzy, is we have more vessels that are going into, let's have our afferents. We have more vessels sending lymphatic fluid into the lymph node and fewer vessels that take the lymphatic fluid out of the lymph node. So because we have more vessels dumping lymphatic fluid in, we have more afferent vessels than efferent vessels, we are going to have an increase in pressure. We're also going to slow down the rate of flow. As we slow down this rate of flow, we are going to give our lymph nodes time to filter out the pathogens. Filter out pathogens. The specific tissue type in our lymph nodes that acts like the filter is our reticular tissue, the reticular connective tissues. And within this filter, made of reticular connective tissues, so when we think of the filter, I want you to connect that to the reticular tissue type. And we talked about this briefly during our tissues lab when I had you compare loose, irregular connective tissues to each other. Of those, we have the areolar connective tissue and the reticular connective tissues that fall into that category. So we use the reticular connective tissue as the filter. You can think of it kind of like a cheesecloth.
A cheesecloth?
Yeah.
Is that like the wrap around like the brown cheese? Is that what you mean by cheesecloth?
Yeah, generally speaking. I know my parents will use cheesecloth when they make homemade yogurt. And they'll take the yogurt, dump it into the cheesecloth, and the liquid, the whey, will filter out, and they'll make their own Greek yogurt that way. In our bodies, we have this very loose connective tissue, this reticular tissue that acts like the filter of our lymph nodes. So the reticular tissues are made of reticular cells. Within this reticular tissue, we're going to have some really big white blood cells that swallow bacteria. Izzy, do you remember the term from lab, our first lab activity? What's the technical term for a cell swallowing a solid substance? Phagocytize. And if we look at this term, we have a macrophage. Macro meaning big, phage meaning to eat. Quite literally, a macrophage is the big eater cell. So when you think of a classic white blood cell that's swallowing a bacterium, think of a macrophage. So within the filter of our lymph nodes, within the reticular tissue that's collecting all the bacteria and filtering it out, we have lots of cells designed to phagocytize the bacteria. We also are going to have those lymphocytes. And what were the two lymphocytes we talked about? Leukocyte's kind of an umbrella term for white blood cell.
Okay.
But then we have the lymphocytes, and there's some that are matured in the thymus, some in the red bone marrow.
Oh, so you got your T cells and your B cells.
T cells and B cells, excellent. So we have our T cells and or B cells also in our lymph nodes. Go for it.
All right, let's see, where do we have a heart?
So here's our lymphatic tissue, or a lymph node. We'll have lots of afferent vessels flowing in. We have fewer efferent vessels flowing out. And within that within that lymph node, we have lots of reticular tissues. Those reticular tissues are loose and will serve as the filter that captures the bacterium. Any questions about lymph nodes?
Sorry.
So there's a heart right next to those legs you can grab. That's on a wooden pedestal. Right there.
Here? Oh, okay, thank you, sir.
No problem.
Lovely. Okay, I will try to bring it back intact.
Sounds good. Our next lymphatic tissue
My heart.
is the tonsil. And we have three groupings of tonsils. We'll talk about these when we get to the respiratory system lab. We'll identify these in the lab. But right now, I want to mention them because they're part of the lymphatic, these are secondary lymphatic tissues. We have the palatine, lingual, and pharyngeal tonsils.
Secondary.
Lymphatic tissues. So we're currently talking about secondary lymphatic tissues. We have the lymph nodes, we have the tonsils, we'll have the spleen, and to the lesser degree, the appendix. So we have the palatine tonsils. When you think of palate, I want you to think of your mouth. So these palatine tonsils are in our mouth, also known as our oral cavity. To find the palatine tonsils, you need to go to the posterior margin of our oral cavity. What does that mean? That means the back of our mouth. These are the tonsils that are most often infected because of their location, and because they're most often infected, these are the tonsils that are very often removed. We have lingual tonsils. When you think lingual, I want you to think of the organ that we use for language. That is our tongue. So lingual has the root word for language in it. That is going to be associated with our tongue. The lingual tonsils are very infrequently removed. They typically don't cause a lot of problems for us. And then finally, we have the pharyngeal tonsils. The pharyngeal tonsils are a mass of lymphatic tissue in the pharynx. Most people don't call them the pharyngeal tonsils, though. Most patients will refer to them as the adenoids. That's the old school name that's being phased out right now.
Pharynx, isn't that a tube?
The pharynx is our back of our throat. It's the part of our throat that has both air and food going through it, as opposed to the larynx, which should only have air going through it. You definitely don't want food going down your larynx. That's called choking. Our pharyngeal tonsils, or adenoids, are often removed because they impede the flow of air and cause significant pain when they swell up. On our tonsils, we are going to have a structure known as a tonsillar crypt. This tonsillar crypt is just an indentation in the tonsil. And what happens in these indentations in the tonsils is that bacteria or any pathogens that may be present in our oral cavity or our pharynx will get moved down into the tonsillar crypt and absorbed into our lymphatic tissues so that we can then filter it in through our lymph tissue. We're also going to have high concentrations of lymphocytes in our tonsillar crypts. So these lymphocytes are the T cells and the B cells. These are going to be the cells that help activate the immune system. More on those when we get to physiology. A lot more on those when we get to physiology. Our next secondary lymphatic tissue or organ is our thymus. Our thymus is going to be located around the trachea. Do you remember the common term for trachea?
The term?
Most people won't say trachea. Instead, they'll say windpipe. Our thymus in particular wraps around the lateral margins of the trachea. So it kind of hugs the trachea on the left and right side of the trachea right before the trachea branches off and goes into our lungs. So it's going to be in our thoracic cavity, right about here where I'm holding my hand, kind of about at the level of my heart. The thymus is broken up into two lobes, one on the left side of the trachea, one on the right side of the trachea. And within these lobes, we have an outer layer and an inner layer, a cortex and a medulla. What other organs have we talked about that have cortex and medulla?
Kidneys.
Our kidneys, and think of our endocrine function. It activates our sympathetic nervous system, secretes adrenaline.
Thyroid. It secretes adrenaline.
Yep.
Keep going.
The adrenal glands.
Well, that's part of the kidney.
Not necessarily. But the kidneys do have a cortex and medulla. The adrenal glands have cortex and medulla. Our thymus also has a cortex and medulla, an outer layer and an inner layer. We're going to have more reticular cells in our thymus, these reticular cells form a barrier that help to connect the bloodstream to the thymus. Within this barrier, we're going to isolate immature T lymphocytes, or T cells. What are the two kinds of lymphocytes again, Izzy? T cells and B cells. Excellent. So when I say developing T lymphocytes, these are T lymphocytes that are not immunocompetent yet. They haven't been fully activated. We're also going to have some hormones secreted from our thymus. One of them is going to be thymopoietin. We're also going to have thymolin and thymosins. I do not want you to memorize the names of these hormones. Do not memorize. What I want you to know is that we have special hormones in the thymus that help us develop T cells. When we are first born, our thymus, relative to the size of our body, is enormous. As we get older and older and older, the thymus, relative to the size of our body, gets smaller and smaller and smaller. Once we hit the age of 14, our thymus not only stops growing, but it actually starts to shrink. In elderly patients, the thymus is almost completely replaced with fatty tissues and connective tissues. This is one of the primary reasons why you are going to see warnings for elderly people to receive certain vaccinations or to avoid certain situations, because their thymus has been completely replaced with fat, or it's been completely involuted. Why would a young child be at risk? They have a ginormous thymus, relatively speaking.
Be at risk for what?
Be at risk of pathogens, because you'll often see the warnings for young children or elderly adults.
Developed their antibodies yet?
Bingo. The young individuals haven't had as much time to develop all of their secondary immune responses. While we're on this topic of secondary immune responses, I want to mention that young people, when I say young people, I mean infants, are generally speaking exposed to 150 to 250 new antigens every day. When they receive a vaccination, they're usually going to be exposed to 5 to 10 antigens in that vaccine. So unlike the new approach that anti-vaxxers are taking right now that I've seen, is instead of saying vaccines are bad, they cause disease, now people are saying, okay, fine, vaccines don't necessarily cause disease, but we need to give vaccines to people later in life because their immune system's not fully developed yet to receive the vaccine. That is a misnomer. When we're first born, we have that very large thymus, and it's handling hundreds of new antigens every day. 5 to 10 new antigens from a vaccine is not going to overwhelm the infant's immune system. So, relatively speaking, if we look at a newborn child, the thymus is almost the size of the heart. It's huge. In an adult, our thymus is much smaller. Any questions? Okie dokie. Well, let's pause for a moment. Why would an infant need to have such a large thymus? Structure equals function. There's got to be a reason it's so big.
Well, the thymus, I mean, that takes in, the function of a thymus is to, like, fight off.
Activate or mature T cells, to make T cells immunocompetent.
Okay. So what's the reason that it's so big?
one of the main reasons the thymus is so big in an infant is that infant hasn't been exposed to most of the pathogens in the world yet, in their sphere or their little bubble that they inhabit. So because the infant hasn't been exposed to most of the pathogens yet, they have lots and lots of T cells that they need to activate or make immunocompetent. By the time we hit adulthood, we've been exposed to most of the pathogens that we're going to be exposed to in the world. So our thymus doesn't need to work as hard.
So that's like natural. Like, here's your thymus, and then eventually it's just going to be little. I mean, that's natural.
That's very, very normal, yes.
Okay. The function of a thymus is to
Activate T cells. I can't get much pithier than that. So if we look at a thymus, we'll have some special tissues on the outside and different tissues on the inside. We have the cortex and the medulla. We're also going to have some lobes and indentation in the thymus. Let's talk about our next secondary lymphatic organ, our spleen. Our spleen is a multifunctional organ. It's kind of a jack of all trades. Our spleen, if we look at it, has a ton of functions. It makes blood in the fetus. It's also going to be a backup system of blood. It's our blood reserve or blood reservoir. So when we are bleeding out and we start to have a decrease in red blood cells in our body, we can take red blood cells that we've been storing in our spleen and dump them into circulation to help compensate for the ones that we're losing as we're bleeding. We also recycle red blood cells in our thymus. And then finally, not our thymus, our spleen. And then finally, our spleen aids us with our immune response. We are going to filter the blood to help us detect any antigens, anything that shouldn't be in the blood, any antigen. Izzy, have I explained to this anatomy class what an antigen is yet? No, it is not, but it's closely related to an antibody. An antigen is anything that will stick to an antibody. So we can have foreign antigens or self-antigens. A self-antigen would be something in our bodies that belongs there that will stick to an antibody. A foreign antigen is something that doesn't belong in our bodies, that sticks to an antibody.
Self-antigen belong?
Yes, foreign do not. So generally speaking, foreign antigens are associated with infection. And if we have self-antigens that have antibodies sticking to them, that's going to be associated with an autoimmune disease. Now our body's making antibodies that are attacking our own tissues. If we look in the spleen, we have two tissue types in the spleen. The red tissue, the red pulp, and the white pulp, or the white tissues. The red tissue has red blood cells, erythrocytes in it. The white tissues have white blood cells, or leukocytes in them.
Red blood cells, what's that called?
In the red pulp.
Leukocyte is white. Blood cells.
Leukocyte is white.
And what's the red one?
Erythrocyte. So if we look in a cadaver, or in the human being to find the spleen is going to be directly above the left kidney. Oh, excuse me, this is a posterior view. The spleen is going to be directly above the right. Oh, I'm getting my left and right mixed up. Yeah, the left kidney is for our spleen. So the spleen's above our left kidney. Another way of thinking about it is the spleen is right by the stomach. I need to get our frame of reference.
Actually, on our body, the spleen is directly above our left kidney.
Mm-hmm.
On this picture.
Both.
Oh. Really? It's above our left? Are you sure?
Yep. Here's the kidney. There's the spleen right above it. That's okay. It's important to get a frame of reference. And if we look at the histology or the tissues in the spleen, you can see that there's some whitish tissue and some reddish tissue, the white pulp and the red pulp. Here's our frame of reference right here with the liver. Our liver is always going to be on the right side. Except for those very few exceptions, there's some people who have a disease that causes their liver, stomach, and spleen to be flip-flopped on the different sides of their abdominal cavities.
Liver's on the right?
Liver's on the right side in most adults.
That's where I was getting confused. That's why I was confused.
Spleen's on the left. So let's finish up with our leukocytes. There are 5 leukocytes that I want you to learn from this presentation. Those 5 leukocytes include the neutrophils, the lymphocytes, the monocytes, the eosinophils, and the basophils. I want you to know what their function is, and I want you to be able to recognize what they look like. So if I show you a picture of one of these white blood cells, you should be able to know which one I'm showing you a picture of. I also want you to know the leukocytes in order of concentration. What's the most common leukocyte? What's the least common leukocyte? To help you remember them in order of concentration, there's a little rhyme. The rhyme goes, never let monkeys eat bananas. And if you never let a monkey eat a banana, you can keep these in order from most common to least common in our bodies. So the most common leukocyte, the leukocyte that's at the highest concentration, is the neutrophil. And the leukocyte that's the hardest to find in the body, that has the lowest concentration, is the basophil. And what's the rhyme again, Izzy?
Never let monkeys eat bananas.
Bingo, never let monkeys eat bananas. Now, if we look at the flow chart for blood cells, if we start off with our hemocytoblast, or our multipotent stem cell that turns into blood cells, this one stem cell can turn into a whole bunch of different kinds of blood cells. Some of them are red blood cells, some of them are white blood cells, some of them are thrombocytes or platelets. We are going to focus on these 4 right here. And it's kind of hard to read right here. Here we go. Our lymphocytes. Oh, excuse me, that's this one right here. So those are the 5 that we're going to focus on. So our first one, or I should say first, we could take these 5 leukocytes and break them up into 2 categories. We break them up into 2 categories based on whether or not they have granules in them. And then when you think of a granule, Lizzie, I want you to think of polka dots. So we have the granule or the polka dotted white blood cells, or we have the agranules or the non-polka dotted white blood cells. These granules or polka dots on the white blood cell are going to contain specific chemicals. There are going to be lots of little vesicles that contain chemicals specific to the function of that white blood cell. Every white blood cell that has granules in it is going to have phil at the end of its name. So the neutrophil, the eosinophil, and the basophil are all granule sites. They all have those speckles or polka dots. Conversely, all of the white blood cells that do not have those granules or do not have those speckles in them are going to have cyte at the end. So we have the lymphocyte and the monocyte that are both agranule sites, or without polka dots. Any questions about these two big categories? Can you see the speckles really well between the granule sites and the agranule sites in this diagram? You have great eyes. I always struggled with seeing them.
So your neutrophil, that's going to be the speckled one. That's going to be has granulocytes.
Yep, so we have lots of little speckles.
Oh, okay. So I guess I was looking. These are polka dots. So I'm like looking for like the dots. Like,
Oh, were you looking at these big things?
Yeah. I'm like, yeah, I can see them.
Those big, I'm glad we brought this up. These big dark purple things are multi-lobed nuclei.
Okay.
So those are not the granules. You can kind of see, if we look right here, you can see some individual granules, individual little spots in that basophil. If we look at this eosinophil, it has a bilobed or two-lobed nuclei. And the basophil also has a multi-lobed nuclei. Our lymphocyte has one giant nuclei. And then our monocyte has a very large nuclei as well. So let's talk about the single most common one. A neutrophil, generally speaking, has a nucleus that has 3 sections to it. It has a trilobular nuclei. It's also going to have lots of individual little granules in it because it's a fill. You could think of a neutrophil as the kamikaze pilot of our immune system. It has one job and one job only. Its job is to get onto the scene of infection and kill the bacteria as quickly as possible. During this process of killing the bacteria as quickly as possible, the neutrophil also kills itself. That's why I like to think of them as the kamikaze pilot of our immune system. We will have some phagocytizing of bacteria in our neutrophils, but what I really want you to focus on with our neutrophils is the fact that they create the killing zone. This killing zone is a small area around the neutrophil where we're going to have the liquids contained in these granules released to the extracellular environment. The granules will exocytize their chemicals. And while those granules are exocytizing their chemicals, those chemicals cause what's known as a respiratory burst. These chemicals are very toxic, and they include superoxide, hydrogen peroxide, and hydrogen chloride. All 3 of these chemicals can be used to sterilize or kill bacteria. The problem with releasing those chemicals to the extracellular environment is the neutrophil kills itself in the process, in addition to any bacteria that are nearby. Our neutrophils are also the first on the scene. In many cases. And this is primarily because of the very high concentration of neutrophils in our body. Think back to when you were in middle school or just entering puberty, that wonderful time of life. Most of us had pimples and acne that we had to deal with. And when you pop a whitehead and that pus comes shooting out, that pus in the whitehead is mostly dead neutrophils and dead bacteria, particularly Staphylococcus. Staphylococcus. Very common bacteria in our pimples. Our next leukocyte that we'll focus on is the eosinophil. It's also a granulocyte. You can see the little granules in it. These granules of the eosinophil are going to have a red or pink color to them. The name eosinophil, or the namesake of eosinophil, is actually the staining agent that colors them red. Eosin dye will stain the granules in an eosinophil. What do eosinophils do for us? Our eosinophils primarily are associated with anti-parasitic effects. So if we have a multicellular parasite in our body, those eosinophils, or the white blood cell, that are going to focus on that parasite. Our eosinophils are also capable of phagocytizing anything that has antibodies stuck to it. So if we have antibodies sticking to an antigen, that's going to be putting a big neon sign that starts blinking over that molecule, and it will tell the eosinophil to swallow that molecule or swallow that cell. Our eosinophils also have enzymes that block excess inflammation and limit the action of histamine. And that's a good thing because eosinophils also activate two cell types that cause inflammation and release histamine into our bodies. So if we have an appropriate immune response, when the basophils and mast cells are activated, we'll also have eosinophils activated at the same time to limit their effect. When somebody has an allergic reaction, the basophils and mast cells are activated, but there are no eosinophils to temper their response. Why? Most allergens are not parasites. Our next leukocyte is another phyll. It is the basophil. Our basophils are the kind of the coordinators or the helper cells that we're going to focus on today. They make it so that other white blood cells can move around with more ease or easier, and so that those other white blood cells can be more responsive. A basophil has lots of dark blue granules in them. So when you think of basophils, I want you to think of dark blue granule sites. If we flip back a couple slides, we have the neutrophil, which has very lightly colored granules in it. The eosinophil has lots of pink granules in it. And the basophil has dark purple or dark blue granules in it.
Are we going to have to look these up in a microscope in this test?
Nope. But I may give you a picture of them for the lecture exam that you will need to identify. All right, let's move on to our A granule sites. The first A granule site that we'll talk about is the monocyte. Our monocytes turn into a macrophage. Now, is a macrophage a big cell or a small cell?
It's a big eater.
It's a big eater cell. So if we think of the monocytes, they're also going to be large cells. A monocyte is one of the largest cell types in the body. And it will turn into the macrophage, so when we activate a monocyte, the monocyte will start to actively phagocytize pathogens that are in our bodies. Some monocytes are stuck in specific organs of our bodies, or tissues of our body. For example, we have one cell type known as a dendritic cell. This dendritic cell is a special kind of macrophage that's in our skin, in our mucosal linings, and also in our lymphatic organs. We also have a special kind of monocyte in our central nervous system, known as the microglial cell or microglia cell. We have another kind of monocyte that's stuck in our lungs called the alveolar macrophage. And then we have another monocyte that's stuck in our liver that's known as a hepatic macrophage. And this name right here is the new school name on the block. The old school name was Kiefer or Knüfer cells. I can't even pronounce it. It's an old German name or German word. Hepatic macrophage is a much more intuitive name because hept is a prefix for liver. It is. So if we think of the hepatic vein, the hepatic portal system, the hepatic artery, those are all blood vessels that go to the liver. A hepatocyte is a liver cell. A hepatic macrophage is a macrophage that's specific to the liver. So when you think of monocytes, I want you to think of the cells that turn into macrophages. And they're all over our bodies. And then finally, our last white blood cell type is the lymphocyte. As a refresh my memory, what are the two primary lymphocytes?
T cells and B cells.
T cells and B cells. There's one that I haven't mentioned yet that makes a small minority of our lymphocytes. That is the natural killer cells. So our lymphocytes will be turned into, this precursor lymphocyte will turn into a T cell, a B cell, or a natural killer cell, an NK cell. T cells are maturated in the thymus, B cells in the red bone marrow. Our natural killer cells, we are not going to spend any time talking about, other than to say that they're super cool. They're my favorite one. And you can take phys to learn more about the natural killer cells. Any last minute questions before we call it quits on this presentation?
I don't think so. Is that study guide going to be ready?
The study will be ready by the end of the week. How's that for an ambiguous answer? All right, well, we'll call her quits then.